It's a pleasure to me introduce Rebecca Lynch, who is assistant professor at the George Washington University and work on isolation of broadly neutralizing antibodies against HIV. And today she will talk about using antibodies against HIV. So, Rebecca.
Thank you so much for that nice introduction. So obrigada. Thank you so much for the invitation to speak. It's so wonderful to be here, and I learned so much. And I just wanted to mention that last time I was here last year, as Gordon says, I had a parasitic infection that I'm no longer infected with, but this year was not completely devoted to science. So I'll start with some background on broadly neutralizing antibodies, and some other people have talked about them, and some of these slides overlap with what Rick Kaup so nicely talked about the other day, but I'll make slightly different points. And so just to start off with, the concept of using passively infused antibodies has actually been around for hundreds of years. It's a very old concept. And here's a slide from a review by Barney Graham, who spoke last week, and this is out of date already. This is from 2015, but the point is that there's been a lot of technology in the past 20 or so years helping us isolate more and more monoclonal antibodies, and as that technology has developed, the use of monoclonal antibodies for treatment has really been useful in a lot of other fields, but not so much in infectious disease. And just in the past couple of years, that's really changing. So I think it's an interesting time to be thinking about how do we use monoclonal antibodies in the context of infectious disease. And so why would we even want to think about antibodies for treatment? And so this is just a reminder of how antibodies differ from some other drug mechanisms, and that they really interact with the immune system. And so antibodies, in the terms of virus infection, can neutralize the virus and interact with the virus, but they can also interact with infected cells. And so this is from a review that's actually talking about cancer. And so in this example, the antibody is rituximab, and the quote-unquote infected cell is a cancerous B-cell lymphoma, and the antibody targets CD20, and it's been very effectively used in cancer to target B cells. And basically, there are a lot of different mechanisms of antibody effector functions, and the constant region of the antibody can interact with all these different cells. And so you can have antibody-mediated phagocytosis, you can have antibody-mediated cellular cytotoxicity, and you can also involve complement. And there's also been shown that immune complexes, antibodies, when they bind to their cognate antigen, form these immune complexes that can actually boost the immune system by increasing antigen presentation by dendritic cells. And I'm saying on the slide that it leads to increased CD8 responses, but really it's been shown in different settings, more cancer than infectious disease, but that you can also have increased neutrophils, so it's just increased effector functions. And so not all of these actions have always been explored in the context of HIV. And then just as a reminder to myself as much as you, that it's important not only to study the variable region of the antibody and how it binds to antigen, but also the constant region of the antibody and how it binds to immune cells. And that was really nicely talked about by Dr. Kaup. And so I'm actually mostly going to talk about how antibodies recognize HIV. So how can we use monoclonal antibodies for HIV in terms of treatment and cure? Again, so Rick touched on this, so I'll just briefly go through this, but there are multiple scenarios. So antiretrovirals work very well. There are some scenarios where monoclonal antibodies might be useful, and especially just in general cure strategies where you're thinking about different combinations. Maybe you're trying to boost CD8 cells, maybe you're trying to use a bispecific antibody, et cetera. But the use of monoclonal antibodies could aid in all these efforts. And so no matter which treatment or which strategy you're trying to use to combat HIV, you have to eventually make a decision of what antibodies to use. And that's really a question that I focus on in my lab. So what are the different kinds of antibodies that we have in HIV infection? So here's just a slide talking about what happens during normal infection. And basically, the virus and the antibody responses co-evolve. And so you have, in general, someone that's infected by a single variant, oftentimes, a virus, and then you have antibodies that develop against this founder virus, and the person seroconverts. But then the virus escapes, and so you have all these different variants that eventually form. And so you have a mutant virus here in blue, then you make blue antibodies, and then this goes on and on and on until you have this quasi-species of virus variants in the person, and then you also have the same amount of same amount of genetic diversity that's reflected in the antibody response. So that's what happens normally during infection. And then some people develop what's called a broadly neutralizing antibody response. And so there are two totally different papers that have been published showing the same exact phenomenon. And Rick showed the figure from Nicole's paper, and so I'll show you the figure from Peter's paper. And basically what it is that you can take tons of serum. So this is 117 serum from chronically infected people and test their neutralization properties against 219 different genetic variants of HIV. And what you find is that there's this pretty linear correlation between the number of people who can neutralize a certain number of viruses. And so if you look here at this dotted line, you can see about 50% of people can neutralize about 50% of viruses. And so that follows that if you're looking for people who can neutralize a lot of different viruses, it's a very small number. And so here, if you look, there are about 10% of people can neutralize about 90% of viruses. So when we talk about broad neutralization, we're really talking about this small number of people. And so also when we're talking about neutralization, this is just briefly to go through the assay. Usually people are talking about the pseudovirus assay, and so what this is that you take a plasmid of HIV with an ONV deletion, and you co-transfect it with another plasmid that's expressing your envelope gene of interest. And then you generate these pseudovirions that are replication incompetent. They can enter the cells, and then they have a defective genome. And then you use this reporter cell line that expresses high levels of CD4 and CCR5, so they're very easy to become infected. And then upon successful integration of the virus, they produce luciferase. And so then you can generate these curves and measure how much does a monoclonal antibody reduce luciferase production, or how much does someone's serum reduce luciferase production. And so here is a dendrogram showing the genetic diversity that you can cover because this assay is so high throughput. So this is the genetic diversity of a 174 ONV panel. And so then just briefly, the people, once they're identified as making broadly neutralizing antibodies, you can isolate their antigen-specific memory B cells, and there are various methods to do that. And then basically you just isolate the RNA from these cells, generate a cDNA library, and amplify the BCR, co-transfect the heavy and light chain, and then you have your broadly neutralizing antibody. And so here's a nice visual of all the studies that you can do, or that some groups have done, and this is very labor-intensive and very expensive and very beautiful work, and this is summarized in this really nice review by Penny Moore, where you have a donor, you isolate broadly neutralizing antibodies, you can test these antibodies for function, you can then use the sequences and deep sequence the person's B cell repertoire over time and look for the other members of antibodies that are related, or B cells that are related to the antibodies you've pulled out. You can do the same thing with the virus, where you can deep sequence the virus over time. You can look at how they interact. You can look at the structure of them. You can generate this massive amount of data on how antibodies, broadly neutralizing antibodies, develop in these people. And so what have we learned? We've learned a lot. But there are just 2 points that I want to make, and that we've learned when someone makes a broadly neutralizing antibody response, it's during coevolution in that person, and that person's virus can always escape from the response. And so therefore, there's not necessarily a clinical benefit to that person from having a broadly neutralizing antibody. And so one way to say this is breast is sort of a side effect, or it's sort of random, the development of breast. That's an oversimplification, but it's somewhat random. And Penny says it more eloquently, but that the acquisition of breath is a side effect of the evolution. And so here, just to highlight this point, is some data that I generated in collaboration with Chaim Schramm at the VRC, where I isolated a broadly neutralizing antibody from this person, NAH44, who's been infected with HIV for many years. And the antibody was isolated from blood cells from a 2006 time point. And then we went back and deep sequenced this person's B cells and looked for all the heavy chain variable regions that were related to this antibody that I isolated. And so what you can see here is here's a phylogenetic tree, and it's color-coded by time. So the 2006 time point, I isolated this broadly neutralizing antibody here, and so this branch on the phylogenetic tree is the branch of broadly neutralizing antibodies. But you can see there's an incredible amount of diversification over years in this person. And then what I really want to point out is that if you go 4 years later and look at 2010, this is the branch that's evolving, this is the branch that's evolving, this is a branch that's evolving, but not this one. The breast isn't being selected for in this person. And nor oftentimes does breast represent a dominant response. And so really, one of the points that I wanted to make is that introducing a broadly neutralizing antibody is introducing a new factor into the balance of the person's own antibodies with their own virus. So developing breast is difficult. That probably has more implications for vaccine design, but a way to get around this is to infuse a broadly neutralizing antibody into a person. And so Rick showed this slide, and basically the point I want to make this slide is just to show that there are all these different sites that broadly neutralizing antibodies can target on the virus, and that means that each antibody is really different, and so when we say BNAbs, they're not all the same. And so there are different ways that we can evaluate how the broadly neutralizing antibodies are different and which ones would be most relevant to you to choose in a treatment strategy. And so this is a graph of just some antibodies showing how many viruses they can neutralize on a large panel of pseudoviruses at different concentrations. And so you begin to see differences in these antibodies, and that if you look at the highest concentration of antibody, so 10E8 is of all these antibodies neutralizing the most at this concentration. But you can see PGT121, for example, may not neutralize the most viruses at this concentration, but it's very potent. So if you go down to lower and lower concentrations of antibody, actually it still covers a lot of viruses more than most of the other antibodies. Yeah, so you start to see some different characteristics. And then you can break this down even further, because I'm talking about the large panel of viruses as one lump thing, but really you can look at the data where you're breaking down how well the antibodies neutralize each different subtype. And so this was just pulling data from the CatNap website from LANL, and so basically what they do is compile all the neutralization data that's been published by different groups and put it in one place where you can search. And so I picked out some antibodies that are being developed for the clinic that have all been tested against the same number of viruses, and then split it out by subtype. And again, you can start to see different characteristics. So for example, 101074 is very broad against subtype A, G, and subtype B, and with a pretty good potency. So here I'm showing an IC80. But if you are running a trial in Thailand, say, or in Asia, where everyone's infected with subtype AE, you might not have such luck. So basically, you can see that the breast number is not equally divided between each subtype. And so, 10E8 is not as potent or broad against subtype A1. And then you can see this very dramatically as well with this antibody VRC2625, which is isolated from a woman who was infected with subtype C in South Africa. And this is an antibody that's not the broadest, but when it neutralizes incredibly potently. And then you go down here and look at subtype C. So it's 65% broad, but its IC80 is 0.01, which is incredible. So it's incredibly potent against subtype C. But again, if you're running your trial in North America, subtype B, it's 13% broad. So you're going to get almost no coverage. Okay, so the antibodies have different characteristics, and then I'm just going to go through some of the published trials that have been done, because there are a lot of trials to date now with some of these new broadly neutralizing antibodies. And so if we go back to some of the first trials that have been published with some of the older antibodies, 2F5, 4E10, and 2G12, in combination, these studies showed that this cocktail could slightly decrease virus load or slightly delay virus rebound when the person was on ART and underwent analytical treatment interruption. But virus escape was detected. And so at a very broad level, the summary points sort of still apply to even today's broadly neutralizing antibodies. But I guess what I want to point out is that the more you study and the more you work at it, what you do is try and decrease the amount of virus load and really increase the amount of time that you can suppress the virus, and then slowly increase the amount of time that virus rebound is delayed. Okay, so then there's this body of clinical trials in the past 5 years where antibodies singly have been infused into people who are viremic, so they're not currently on ART. And the latest study was presented at CROI with PGT-121. And so of the ones that are published, here's just a quick overview of what I'm talking about, where you can pull from each of the papers, and basically what you see is that compared to baseline the day of infusion, there's a virus load drop when you infuse a broadly neutralizing antibody, but the virus almost always rebounds, not in every case. So why does the virus rebound? There's sort of two obvious answers to the question, and one is that the antibody is cleared. It's effective, it causes virus load to drop, and it's cleared from the body. Or the other reason is that the virus is escaped and become resistant to the antibody. And I'll say, for the most part, the majority of cases, the virus escapes the broadly neutralizing antibody during monotherapy. Escape is not always the same, and I think it's interesting and important to study how the virus escapes so that you can improve broadly neutralizing antibody therapy in the future. So in general, we can see that some of the differences are that CD4 binding site antibodies tend to select for more resistant viruses that already pre-exist, and maybe this is because CD4 binding site antibodies aren't quite as potent as some of the V3 glycan antibodies. The V3 glycan antibodies tend to induce de novo mutations that appear rapidly, and possibly this is because they require a single nucleotide change, because all you need to do is get rid of the glycan or shift the glycan and the virus is resistant. And really you can accomplish that glycan shift with a single nucleotide change. And so recently this year, there's the first combination therapies published out of the Nussensweig lab. And so this is the study of infusing 3BNC117, which is a CD4 binding site antibody, and 101074, which is a V3 glycan antibody, at the same time into participants who were not on ART. And the overall point here, so this is the virus load dip graph, is that the length of suppression is much longer than when you have a single antibody. So already just by infusing 2 antibodies, you're seeing better results than with 1 antibody. So one point is, does pre-existing sensitivity to the antibody matter? i.e., the viruses this person is already infected with, they can't help or change. What is the effect of those viruses on antibody therapy? And so yes, it absolutely does matter how sensitive to neutralization, in particular, your pre-existing viruses are. And so there's indirect evidence for that, because some of the studies pre-screen people for their sensitivity to antibody neutralization and some don't. And what you can see is that when you pre-screen people, there's better clinical effect. But then we also have some direct proof that this is true. And so I'll show some data from the VRC01 trial that was conducted at the VRC, because this is data I generated. And so we were, what we did was we looked at a couple different time points in people who are infused with a broadly neutralizing antibody, and you can sequence the virus at these different time points, and then you can generate those pseudoviruses that I was talking about and functionally test the virus for how sensitive they are to VRC01. And so here's just a dendrogram showing the pre-existing virus diversity. And so you can see these people are all chronically infected with HIV and have genetically diverse quasi-species already pre-existing. And then when you clone these viruses and test them for function, what you can see is the 2 people who had the least clinical effect, so their virus load was really not that much affected by VRC01, were the people who started with the most resistant viruses. So very obvious, but you can prove it. Interestingly, from the combination therapy trial, these people were pre-screened for sensitivity to both antibodies. And so even though you pre-screen, you, depending on how much detail you go into, it's really hard to completely screen the entire quasi-species. And so basically what they found in this trial is that despite pre-screening, there are some people who started off with resistant virus. And you can separate these people out. And you can see that when you separate people out by being sensitive or resistant at the beginning, that there's a dramatic difference in virus load drop. And so you can see that the length of suppression is much longer and better when you have sensitive virus. compared to when you don't. So 3 individuals had pre-existing resistance to at least one antibody. So in effect, they were having monotherapy. Those with the resistance to one antibody developed resistance to the other antibody. So it's very easy for these people to escape both antibodies. So how does this resistance develop? I'm just going to show examples from one person, but basically this is just highlighting data where we can take from the day of infusion and then as the virus rebounds. And so this is a graph showing virus load here in color, the virus load dips, and then the antibody concentration is here in black. And so you can see that the antibody concentration is decreasing as the virus is rebounding, but the virus is rebounding, the antibody is still detectable. So it's rebounding in the presence of antibody. And then so what we can see if we clone virus from these 2 time points is in almost every single person, the virus becomes a little more resistant. Just resistant enough to replicate in the presence of the antibody. And that even people who start off pretty resistant to VRC01, you can see that there's some still selection pressure by the antibody and they become completely resistant. And so then we went in and looked to investigate how these people were escaping. So there's just one example. That loop length was actually a fairly, I say easy, I think it was easy for the virus, way for this person to escape. And so you can see that if you just graph the length of the V5 loop to start off with, there's a little bit of diversity in the quasi-species, but that as the virus is rebounding, there's selection for V5 lengths that are shorter. And then you can graph the length of the V5 and correlate it with neutralization sensitivity, showing that there's an association and that all the viruses with the shorter V5 loop are actually more resistant to VRC01 than the viruses with the longer loop. And then importantly, I also want to say that this is not in an immunologically naive person. You always have to remember that this person has their own immune response and that this person has their own antibodies. And so even though you're introducing this heterologous antibody into the system and the virus has to shift and become resistant to the antibody, it also has to remain resistant to its own antibody response. And so here I'm showing serum. So this is sort of flipped where resistant viruses are at the bottom of the graph. And you can see that over this month, the viruses remain resistant to also the person's own antibodies. And so you have all this selection pressure on the virus, but the virus is more than capable of dealing with it. So just in general, from all these studies, you can see that there are a couple different ways for resistance to develop to the different antibodies. There's selection for more resistant strains within the pre-existing quasi-species. There's selection for unusual residues within the antibody epitopes. There's selection for different loop lengths, which is sort of an indirect way the virus can affect neutralization sensitivity. And then there are also de novo mutations within epitopes, such as glycan shifting with the V3 antibodies. So what about in the context of ART and cure strategies? So there's also a body of work about analytic treatment interruption where you start with people who are fully suppressed on antiretrovirals. You infuse antibodies into these people, have them take a break from taking antiretrovirals and see if how long the antibodies can suppress the virus in the absence of ART. And again, this is just published from the Nussensweig lab that they've done it with combination 3BNC 117 and 101074. So in general, from these studies, we can see that antibodies can delay virus rebound, and there is genetic restriction. So oftentimes, the virus that's rebounding is more resistant to the infused antibody. So there is some sort of selection pressure that's occurring. And then it's interesting, again, to look at the what happens when you infuse 2 antibodies. And so this is a graph of all the participants from that study. And these are some historical controls showing there's a Kaplan-Meier curve showing the weeks to virus rebound in the absence of antibody in black, with a single monoclonal antibody 3BNC117 in red, and then in blue with 3BNC117 and 101074. And again, these people were pre-screened to make sure they were sensitive to the antibodies to begin with. And so you can see you're progressively getting better. They're increasing the time it takes for the virus to rebound in the absence of ART. But again, despite pre-screening, there are people who did have resistant viruses in their reservoir, and you can separate out the data in terms of who's sensitive and who's resistant, and then again, you can see really big differences in clinical effect here. And so this is just another reminder that when you're starting off with pre-existing resistance in a combination, you're essentially getting monotherapy from which you can escape. And so what they found in this trial, which is, I think, interesting, we'll see what happens in the future, is that 101074 had a longer half-life, a slightly longer half-life than 3B and C117. And so presumably once 3B and 17 was clear, these people were essentially on monotherapy and they all developed resistance to 101074. And that's how the virus presumably rebounded. And then this is also to remind us, this is a poster at Croy from Katie Barr's lab, that you also have autologous antibodies. So just because people are on ART doesn't mean your antibody response goes away. It's there. And in the context of virus rebound, the antibodies are still driving evolution. They're neutralizing the virus, and the virus has to escape. And so what do we know about antibody responses on ART? It was a little difficult to pull together all the data because people define antibodies differently. But overall, I would say that there's a general consensus in the literature that antibody titers decline once you're on ART and there's not actively replicating virus, but they're still detectable. You still have a serum response there. And that titers are higher the longer time you've been infected before you go on antiretroviral treatment. So this is all a really long introduction to say that some of the questions that I'm really interested in my lab, and this is preliminary work, we don't have answers, but I'm really interested in how the complexity of the virus reservoir relates to antibody sensitivity. And so here's my graphical abstract. How long you have been infected before you start ART affects the diversity and complexity of your virus reservoir. And then how does that affect your autologous antibody response and your sensitivity to bNAb treatment, bNAb therapy? And these, I think, are all important questions to think about when you're thinking about enrolling people into different cure strategies that are going to rely on either the person's antibody response or a broadly neutralizing antibody to work. And so I'll show some of our preliminary data. And so this is all work that we've done in collaboration with Brad Jones's lab. And so all the work I'll be showing you is 8 participants. So the Jones lab has developed this cohort of people who are enrolled in a clinic in Toronto. So these are people who are mostly infected with subtype B virus and have access to antiretrovirals. And so here are some clinical characteristics of these participants. They're all male. They're all pretty well suppressed, infected with subtype B virus. And there's some variability in the time that they were infected before they went on ART, or the time they were diagnosed, before they went on ART, and how long they've been on ART. And also differences in the reservoir. And so here's the infectious units per mil over here that's measured by the Jones lab. And so how do they measure the reservoir size of these people who are on suppressive ART? They do virus outgrowth assay, but importantly, they have limiting dilution replicates. So basically, they can take leukapheresis samples from these people, isolate their resting CD4 T cells, plate them in limiting dilution replicates, and activate them, and wait for virus to grow in a certain number of the wells. They measure virus growth by P24, and then here's just a little cartoon schematic of the results where you can see at different dilutions of CD4 cells, you can estimate from how many wells are positive the size of the reservoir. But a side effect of doing this assay is that you're also generating all these samples of the person's reactivated virus, which is really useful for us in our neutralization assays. So all the work we're doing here is not on pseudoviruses, but actually on the person's own reactivated virus from their inducible reservoir. Right, yeah. So we were interested in getting these supernatants from the Jones lab, and the first question we were curious about is how sensitive are these outgrowth viruses to different broadly neutralizing antibodies. And so we put together a panel of antibodies that are either in clinical trials or mostly being developed for clinical trials, just in antibodies that would be representative of targeting different epitopes on the virus. And then we ran large panels where we had a couple different outgrowth supernatants from each participant and then ran them against this panel of 16 antibodies. And so this is just to show you the large amount of data we generate, where you can see we have these neutralization curves for every virus and antibody pairing. And this, in each column, is the antibodies are graphed by their target epitope, and then there are about 3 or 4 antibodies from each group on each graph. And so you can start to see these different patterns of how well the antibodies neutralize each. Oh, sorry, and the rows are different virus outgrowths. This is from one participant. And then this is a summary slide showing the IC80 data from all these results. And so basically, each participant is in a black box, and then the number of viruses are in the rows, and the antibodies are in columns, as I said before. And then the IC80s are colored just according to potency. And so we can start to see differences. So I guess I'll first start with the antibodies. We can see that in general, if you look at the CD4 binding site antibodies, the first thing you can notice is VRC01, not the best antibody. So it's one of the first to go into clinical trials, so there's a lot of data on it. But it's not as potent or broad as some of the antibodies that have been isolated subsequently. So you can see VRC07, 523, 3BNC117, and N6 are all sort of better in terms of they neutralize more viruses more potently. But they're not super potent. Whereas then if you switch to the V3 glycan column, you can see they sort of either neutralize or don't neutralize. And when they neutralize better, more potent than the CD4 binding site antibodies, but they're also it's easier to find resistant viruses. And then from the virus side, you can see that there are differences. So I'd say overall, if you just look at people, there are some people whose viruses are fairly sensitive to a lot of the antibodies. And then there are some people whose viruses are pretty resistant to a lot of antibodies. And then you can also start to look in each participant and see, so I will say these are only 5 to 6 outgrowths that we're testing. So this is underestimating the diversity of the inducible virus reservoir, but I'll also say that 5 to 6 viruses is sufficient, where you can also start to see differences, and you can start to see the diversity even within these 5 to 6 viruses. And so you can see that this person that I pointed out, they seem pretty sensitive to most of the antibodies, but you look at 101074 and you can see there's already one virus that's resistant to this antibody, and there's one virus that's already resistant to PG9. And so I think that's a really important point to think about when you're going to use broadly neutralizing antibodies as therapy. And then you can also see that there's some people who just have more diversity in their reservoir, and so this person, half their viruses are resistant to 3BNC117, half their viruses are resistant to 101074, another half are resistant to the V2 antibodies, but some of them are super potently neutralized by this VRC26 antibody, and so you can see that there's a lot of genetic diversity within these people's reservoirs, and we're sequencing all the viruses, but we don't have that data yet. And so the next question we are interested in is, what about the people's own antibodies? And so this is a little bit harder to do. So normally I just like to put people's serum into a neutralization assay, but because all these people are suppressed on ART, we can't do that. And so what we did is take plasma and isolate their IgG away from the antiretrovirals, and then we normalized IgG concentrations in these downstream studies. And so the first thing we did was just ask, do they have HIV antibodies? And so here's a graph showing their binding titers to GP120. And so you can see that everyone has detectable HIV binding antibodies. There is diversity in the titers, but they're all detectable there. We next asked if these antibodies could be neutralizing. Do people have neutralizing antibodies? And so the first thing we did was test on this pseudovirus panel that's a test panel that was published by Alan DeCamp, and so it's supposed to be representative of global genetic diversity and also global neutralization sensitivity. So basically what this paper did was look at large panel data and then try and scale that down, that if you test antibodies or serum on this much smaller panel that's much easier to do and uses less reagent, you'll get a somewhat relevant indicator of how broad or potent that antibody or serum is, and to try and standardize some of the neutralization testing that people do in different groups. So what we see is these people are pretty normal, I would say. So we see low-level neutralization of a bunch of different viruses. So these people have HIV antibodies despite being on ART, and most of it isn't potent. So these are not people who are the elite neutralizers, but they all have neutralizing antibodies. And so that is important. But then the real question we're curious about is, can they neutralize their own reservoir virus? So basically, if the reservoir is an archive of all the viruses this person has been infected with, presumably some of the viruses are from earlier in infection and would be neutralized by the person's own antibodies. But if the virus reservoir is more closely representative of the virus diversity at the time of ART, then presumably most of the viruses will be resistant because of antibody and virus coevolution. And so I was interested in this question because sometimes you think about strategies for cure, such as boosting NK cells, that would really be enhanced if antibodies could be incorporated. And I think a big question is, are the person's viruses all resistant to their own antibodies or not? And so we're just starting working on this. We don't have the answers yet, but I'll just show some of our preliminary data. And so we're testing all the participants' virus outgrowths against all their IgGs. And then I highlighted the autologous antibody virus interactions in orange. And so what we can see overall is that not everyone is completely resistant, but they mostly are. And so we'll see how this goes as we add an increased number of N. But so this person, you can detect neutralization of autologous virus, but I'm not sure what that means in vivo. It's not very potent, so I don't know if this is strong enough neutralization to exert effects in vivo, so we'll see. But I guess just preliminarily, we don't see any viruses that are very sensitive to their own antibody neutralization. And then overall, we also see some interesting phenomenons where this person's IgG can bind to HIV, can neutralize pseudoviruses, but it doesn't neutralize any viruses from these participants so far. And so the sequencing data will actually really help to see how closely related all these people from Toronto, their viruses are. Maybe I don't know what this person's antibody, what epitope they're seeing. And then we can see that this person's antibody can neutralize some of these viruses better than other people. So there does seem to be some functional differences that we would like to explore in the future in how strong the autologous antibody response is in the antibody response while these people are on suppressive ART. So in conclusion, the virus can escape from broadly neutralizing antibodies, even when people are started from suppression. So they do not have theoretically actively replicating virus. But it is harder to escape from 2 antibodies. And we can see that quite clearly from the nuisance flag papers that were published this year. Virus populations that become more resistant to broadly neutralizing antibodies also have to maintain resistance to autologous antibodies. So there's always that selection pressure still in the person from their own antibodies. And then finally, second to last point, is that the complexity of the virus reservoir will really affect how likely it is that people have pre-existing resistance to antibodies. And so it's important to think about that and try and incorporate that into any cure strategies using broadly neutralizing antibodies. And then finally, I think it's also really important to study autologous antibodies. And so we don't have much data yet, but so far what we see is that no one can potently neutralize their own virus reservoir. So I think that's also an important factor to consider. So I'd like to thank everyone. All the clinical trial data was from my time at the VRC, and then all the data on people who are on suppressive ART are participants from the Maple Leaf Clinic, and is a collaboration with Brad Jones's lab, who recently moved to Cornell. And then I'd like to thank my lab. Most of the work was done by my graduate student, Andrew, and Michelle is a postdoc from Rio who just started in my lab, and so I met her at this conference, and so that's been really exciting. So thank you.
Thank you, Rebecca. Now we have time for some questions.
Thanks for a really excellent talk. One question I have is, you've looked at all the neutralization and all the function from the systemic point of view, but a lot of what we think about is at the tissue level and in lymph nodes, and we saw some of that data in terms of Adrian's work and biopsying lymph nodes and looking. So have you had an opportunity to ask these same questions and think about what is the accessibility of antibody to tissue? How well does it get in the patient? And where is that from a functional perspective at the lymph node or even the gut level, since those are really the sites where the virus lives?
I think that's a really important question, and I have to say that for a long time I didn't think about it correctly, and I sort of assumed antibodies go everywhere, they're circulating, so looking in the blood is good enough, but thanks to people like Tom and Adrian's work, I think that's actually a really important question, and so then the question is collaborating with someone who has those kind of samples to look at, because I think that'd be really interesting, and we haven't had a chance to do that.
Thank you.
So you've been looking at neutralization
Yes.
by autologous antibody, and not too surprisingly, kind of variable.
Yeah.
All right, but all these antibodies should bind envelope. And so how much of a role do they play in ADCC, and how do they interfere with earlier efforts that we saw to augment ADCC by addition of a single monoclone?
So that's also a really good question. It makes me realize that I did not include some data from the paper we just published with Brad's lab. And this is not looking at autologous neutralization, but we looked at the ability of the same broadly neutralizing antibodies in the neutralization panel of outgrowth viruses to bind to matched infected cells. So these are cells not infected with, not in vitro infected, not peptide pulsed, but presumably presenting envelope protein in a more physiologically relevant conformation. And what we saw was that basically neutralization correlated with binding, and so it's not exactly the same thing as ADCC. But so one argument is that I would assume that actually the ADCC function is very similar to the neutralization data that we see, because I'm not sure how different. Like I think if it's escaped from I think if the virion has escaped from neutralization, I suspect that it's also escaped from infected cell binding. But that being said, on the other hand, we are planning on doing it, so we just don't have the data yet. And it's important. It's arguing against the point I just made. It's very important. We're going to look at the subclasses of the antibodies, not just look at IgG, and look at other functions.
For a number of years now, patients have been able to take one tablet a day with 3 or 4 drugs, and that clearly works, although the regular need to do this for a very prolonged period is obviously a downside. So if you had 3 or 4 monoclonals of the kinds of potency that are being worked on now, I would think that they would work very, very well. The question then comes to the translation and convenience. What are the prospects that these could be developed as truly long-lasting formulations that would only be given very infrequently? Because that, to me, seems to be the only true possible advantage of a monoclonal cocktail over a conventional antiviral cocktail.
So I guess I would answer in 2 ways. And one is that I tried to phrase things that I think these studies are important because I think that in different cure strategies. So I think knowing how easy it is for the virus to escape or how the antibodies interact with virus is important to know when you're then designing CAR molecules or DART molecules, or any of these strategies that involve a variable region binding to the virus. So not so much for treatment, but rather actually in cure strategies as supplemental cure strategies. And then to actually answer your question, I guess the answer is I don't know. But theoretically, going back to the first slide, there could be cases like infants where you don't want to give antiretrovirals. And so if you do have some combination of a bivalent or a trivalent molecule that is more potent than the current monoclonals, I think there would be some. I think there's potential.
Yeah, it comes down to costs and benefits. Antiviral pills are relatively cheap to actually manufacture, as opposed to what they're sold for. It's a different thing entirely. And, monoclonals are not cheap when you're making them in these kinds of amounts. And if you're using 3 of them, and if you have to deliver them once every 3 months, that might be a very different scenario than once a week, for example. So, to my mind, I mean, and I'm an antibody guy. I mean, I like antibody. I love the kind of stuff you're doing. But if you're, and of course, I accept what you say in the cure context, which is somewhat different than standard therapy.
Right.
But, to use a standard therapy, it comes down to, is this a better mousetrap? Is this a mousetrap that we can make and use?
Yeah, I mean, on the one hand, I would say that the more we study it, theoretically, we could come up with some cheap molecule to make. On the other hand, I'm not, I'm a basic scientist, and so I can't guarantee that will happen, and you're absolutely right that as of right now, they're not cost effective, but I still think it's important to try and study if there is a possibility.
Yeah, it's in the hands of the manufacturers and the formulation people and the pharmacologists and the non-basic science people like us. But that's where it will come down to. It's not. I
mean, but Sanofi is working on tri-specific antibodies, so presumably some drug companies see some future. And
their downside is the more you engineer an antibody molecule, in general, the shorter its half-life because it's cleared more quickly. So it's a swing and roundabout. But as you say, there's investment in it, and maybe it'll get figured out.
Well, with regard to John's point there, I think there's a lot of room here for basic science investigation. There's not much been done to look at different two-way combinations of antibodies and the results of the virus escaping. So, for example, I think it's possible that for a virus to escape certain combinations of two antibodies, the virus may pay too high a price to do that and may not survive in the in vivo setting where the host already has an antibody response to the virus. And I think it's an area where more work needs to be done, looking at different combinations from the 5 different classes, and are there some two-way combinations that could work in vivo setting that the virus pays too great a price and can't escape.
I couldn't agree more. I think that's really interesting.
So we have one last question here in the applicative. Regarding the use of monoclonal antibody for neutralization of reservoir, do you think that could be better performance if using monoclonal antibody from samples collected on early infection?
Oh, that's interesting. I'm not sure if I understood the question. Is the question, would we find more broadly neutralizing antibodies from earlier in infection?
Yes.
So that's interesting. In general, when people study the development of broadly neutralizing antibodies, usually the length of infection is associated with the development of breath. That's likely for a couple different reasons. Maybe the B cells need to see enough diversity in the epitope. Maybe also, sort of anecdotally, a lot of the people who develop breath are super infected. But then conversely, when people study children, there's some really nice work out of Julie Overbau's lab and then more that children can develop broadly neutralizing antibodies with very little somatic hypermutation. So originally I probably would have said, no, you're not likely to find a broadly neutralizing antibody from early infection because you need to see all this virus diversity and you need to have all this diversity in the antibody side and develop a lot of somatic hypermutation. But based on the work in children, I say maybe that's not true.
So thank you, Rebecca, for your lecture. Thank you. And now we go to the next presentation.